Welcome to the deep dive. Just take a moment and feel your body moving, even blinking, or thinking about running. It's all controlled movement, isn't it? But have you ever really stopped to think, how? How does your body just do all that? How does a thought become an action almost instantly? Today, we're really going to dive deep into that incredible mechanism. Our mission is to explore the mind-blowing details of our muscles and the nervous system that controls everything. And you've shared some fantastic notes with us, a really comprehensive review, which gives us a great roadmap for this. So we're going to unpack these fundamental biological processes. We'll start really small, microscopic even, with what makes a muscle fiber twitch. Then we'll look at those super fast electrical signals in our nerves and finally zoom out to see how it all coordinates for, every single thing you do. We're aiming to pull out the key insights, the surprising connections, make it all make sense. Okay, let's get into this amazing machinery inside us.
Absolutely. And understanding these fundamentals, it's not just, book learning. It's key to understanding so much more. Things like why you get a muscle cramp or how you react so fast to touching something hot. It's all linked. Once you see the connections, it's really quite eye-opening.
Okay, so let's start with the muscles themselves, the movers. Our bodies have three main kinds of muscle tissue, right? Each with a different job.
That's right. We classify them as skeletal, cardiac, and smooth muscle. Each one is uniquely built for its role. Skeletal muscle is probably what most people think of first. It handles all the voluntary movements, picking things up, walking, smiling, you name it. The cells themselves are long, sort of tube-shaped, and interestingly, they have multiple nuclei per cell, multinucleated, and they attach to your bones, or sometimes your skin. Under a microscope, they look striped, or striated. That's a key feature.
And that voluntary control is the big thing for skeletal muscle, yeah. But I saw in the notes some exceptions, like breathing muscles or eye muscles. They seem less voluntary sometimes.
That's a really sharp point. Yes, while most skeletal muscle is under conscious control, take breathing. The diaphragm, for instance, mostly works automatically, reflexively. We can control it, hold our breath, breathe deeper, but we don't have to think about it constantly. It just happens. And eye muscles, they make incredibly fast, precise adjustments all the time without us thinking. But we can also choose where to look, so it shows the system's flexibility.
Okay, interesting. What about the others? Cardiac.
Cardiac muscle is unique. It's only found in the heart. The cells are shorter, branched, and they usually just have one nucleus. A key feature is they're connected by these special junctions called intercalated discs. This helps the heart beat as one coordinated unit. And critically, cardiac muscle is completely involuntary. You don't consciously decide to make your heart beat. Like skeletal muscle, it's also striated, which points to that organized structure needed for powerful pumping.
Got it. Heart only, involuntary, striated. And the third type, smooth muscle.
Right. Smooth muscle. You find this in the walls of hollow organs. Think your digestive tract, blood vessels, bladder, places like that. Everywhere except the heart, basically. These cells have one nucleus. They're involuntary, but they lack those striations. They look smooth under the microscope, hence the name. They handle things like peristalsis, those wave-like contractions, moving food along your gut, or changing the diameter of blood vessels to control blood pressure. Slow, sustained actions, often.
Okay, three distinct types, three distinct jobs. But you mentioned all muscle tissue shares some fundamental abilities, sort of their superpowers. What are those?
Yes, there are 5 core properties: excitability, conductivity, extensibility, elasticity, and contractility.
Excitability. That sounds pretty fundamental. Like they have to be able to receive the message in the first place, right?
Exactly. It's their ability to respond to a stimulus. Usually that's an electrical signal from a nerve or maybe a chemical signal like a hormone. It's the muscle's capacity to listen for instructions.
All right, now this is where, for me, it gets really interesting, zooming right in. We know what muscles do, but how? How does a muscle actually shorten? What's happening at the smallest level?
You're talking about the sarcomere. That's the basic contractile unit, like the fundamental engine piece within the muscle cell. And this little engine sits within the muscle cell, which has its own membrane called the sarcolemma. And inside, there's also a special storage network for calcium called the sarcoplasmic reticulum. We'll definitely come back to calcium. Inside the sarcomere itself, you have these precisely arranged filaments, thick ones and thin ones, made mostly of proteins. Some proteins are contractile, they do the actual pulling. Others are structural, they hold everything in place. And some are regulatory, they control when the pulling happens.
Wow, so it's incredibly organized.
Extremely. And what's fascinating is how this precise layered structure allows for really efficient force generation. It scales up beautifully. You go from these tiny filaments called microfilaments building up into a myofibril. Then bundles of myofibrils make a muscle fiber, which is a single muscle cell. Then bundles of fibers make a fascicle, and bundles of fascicles make the whole skeletal muscle you see. And each level is wrapped in connective tissue, endomysium around fibers, paramysium around fascicles, epimysium around the whole muscle, like layers of insulation and support, channeling the force.
Okay, structure makes sense. Organized layers. Now, the action. How do those filaments actually slide and make the muscle shorten? The sliding filament theory, right?
Exactly, the sliding filament theory. This is the core mechanism. It's like a molecular dance. The main players are proteins called actin, that's mostly the thin filaments, and myosin, the thick filaments. Plus, there are helpers, troponin and tropomyosin, and you absolutely need calcium ions, Cas2 plus amide, and energy in the form of ATP.
ATP, the body's energy money. How does that fit in?
It's crucial. The myosin filaments have these little heads that want to grab onto the actin filaments. But to do that, they first need energy. An ATP molecule binds to the myosin head, and when it's broken down hydrolyzed into ADP and phosphate, that energy cocks the myosin head, putting it in a high-energy state ready to bind to actin. Then, for the myosin head to let go of the actin after pulling, a new ATP molecule has to bind. That binding causes detachment.
So it's like a cycle. Attach, pull, detach, recock.
Precisely. Billions of myosin heads doing this cycle over and over, pulling the actin filaments towards the center of the sarcomere. That's what shortens the muscle fiber.
It's amazing, like billions of tiny rowers pulling on ropes. A microscopic tug of war that lets me lift my coffee cup. That's wild.
It really is a fantastic example of converting chemical energy from ATP directly into mechanical work.
Huh.
But there's a gatekeeper in this process.
Ah, the regulatory proteins. And calcium.
Exactly. Calcium acts like the master key. It actually plays two vital roles, one outside the muscle cell and one inside.
Okay, 2 roles for calcium. Let's break that down.
Right. First, think about the signal arriving from the nerve. When that nerve signal, the action potential, reaches the end of the nerve fiber, the neuromuscular junction, it triggers calcium channels in the nerve ending to open. Calcium ions then rush into that nerve ending from the fluid outside the cell. This influx of extracellular calcium is the signal that tells the nerve to release its chemical messenger.
Which is usually acetylcholine for muscles.
Correct. Acetylcholine, or ACH, that's the first role of calcium, triggering neurotransmitter release from the nerve. Now, second role. Once ACH crosses that tiny gap and binds to the muscle cell, it triggers a new electrical signal in the muscle cell. This signal travels along the muscle cell membrane and dives deep inside through structures called T-tubules. This signal tells the sarcoplasmic reticulum, that internal calcium storage tank we mentioned, to release its stored calcium inside the muscle cell.
Okay, so now we have calcium flooding the inside of the muscle cell.
Yes, intracellular calcium. This calcium then binds to troponin, one of those regulatory proteins sitting on the actin filament. When calcium binds to troponin, troponin changes shape, and the shape change pulls the other regulatory protein, tropomyosin, out of the way. Tropomyosin normally blocks the sites on actin where myosin wants to bind, so calcium binding to troponin is like unlocking the door, moving tropomyosin, and allowing myosin heads to finally grab onto actin and start the sliding filament cycle.
Wow. So calcium is essential both for the nerve to talk to the muscle and for the muscle machinery itself to engage.
Absolutely critical on both fronts. No calcium, no message release from the nerve, and no way for myosin to bind to actin inside the muscle.
Okay, this leads perfectly into the message carriers. How does the brain's command actually get transmitted? You mentioned the action potential and ACH.
Right. It starts with that electrical nerve signal, the action potential, traveling down the neuron. Now, cells generally communicate chemically. The sending cell, the neuron, releases a chemical, a first messenger. In this case, it's the neurotransmitter ACH. The receiving cell, the muscle fiber, has specific receptors waiting for that messenger.
And ACH is the key that unlocks the muscle response.
Pretty much. ACH gets released into the synaptic cleft, that tiny space between nerve and muscle. It drifts across and binds to special channels on the muscle cell membrane. These are ligand-gated Ni plus K plus channels. When ACH binds, these channels pop open. Sodium ions, Na+, which are highly concentrated outside the cell, rush into the muscle cell. This influx of positive charge rapidly changes the electrical state of the muscle membrane, that's depolarization, and this electrical change is what triggers the release of calcium from the SR and ultimately causes contraction.
And how does it stop? We need muscles to relax too.
Ah, good point. There's an enzyme right there in the synaptic cleft called acetylcholinesterase. Its job is to rapidly break down ACH. As soon as ACH is broken down, it detaches from the receptors. The channels close, sodium stops rushing in, and the muscle cell can repolarize and relax. It's a very fast on-off switch.
Incredible. The precision is just staggering. That moment, the thought becomes action.
It really is. Now think about when you really work your muscles hard. They bulge, right? And eventually they get tired. Let's talk metabolism. Skeletal muscles are metabolically flexible. They prefer using oxygen aerobic respiration because it yields a lot of ATP, but they can switch gears.
Why would they switch?
Well, during intense exercise, when muscles contract forcefully and bulge, they can actually squeeze the blood vessels running through them. This compression can temporarily reduce the delivery of oxygen and even glucose via the blood.
Ah, so they cut off their own supply line a bit.
In a way, yes. But muscles store some glucose internally as glycogen. So if oxygen delivery drops, they can switch to anaerobic metabolism, breaking down that stored glucose without oxygen to keep generating some ATP. It's much less efficient, only producing a couple of ATP per glucose molecule compared to aerobics 30-something. And it produces lactic acid as a byproduct, but allows muscle function to continue for a short period during high intensity.
And that's linked to the burn we feel.
Largely, yes. The buildup of metabolic byproducts contributes to that sensation. Muscles also have another quick energy source, creatine phosphate. An enzyme called creatine kinase can quickly transfer a phosphate group from creatine phosphate to ADP, making ATP almost instantly, like a very short-term energy buffer.
Makes sense. Quick bursts of energy. Now, muscles don't just contract in one way, do they? I remember isotonic and isometric.
Correct. We have different types of contractions based on whether the muscle length changes. Isometric contractions are when the muscle develops tension, but it doesn't actually shorten or lengthen. The force generated isn't enough to move the load.
Like pushing against a solid wall.
Yep.
Or holding a really heavy weight steady.
Exactly. You're generating force, you feel the tension, but the muscle length stays the same. Then you have isotonic contractions, where the muscle does change length and moves the load. These come in two flavors. Concentric is when the muscle shortens as it contracts, like lifting a weight, the classic bicep curl. Eccentric is when the muscle contracts while it's lengthening. This is often about controlling movement or resisting gravity, like slowly lowering that same weight back down. It's still contracting, but elongating under tension.
Okay, that distinction is important. Lifting is different from lowering slowly.
Got it.
Yeah.
Before we move to the nervous system, I found the naming convention for muscles fascinating. It's like a code.
Oh, it absolutely is. The names often tell you a lot about the muscle. You get clues about the direction of the muscle fibers, like rectus means straight, parallel to the midline, transverse is perpendicular, oblique is diagonal.
And size. Maximus, minimus, longus, brevis. Right. Maximus for largest, minimus smallest, longest long, brevis short. Latissimus widest, vast is huge, magnus large, major larger, minor smaller. Size clues are very common.
Shape too, right. Deltoid is triangular.
Exactly. Deltoid for triangle, trapezius like a trapezoid, rhomboid diamond-shaped, orbicularis circular. The shape is often right there in the name.
And action. Flexor, extensor, abductor, adductor.
Flexor bends a joint, extensor straightens it, abductor moves a limb away from the midline, adductor moves it closer, levator raises something, depressor lowers it, supinator turns the palm forward, pronator turns it backward.
Number of origins, biceps, triceps, quadriceps.
2, 3, or 4 origins, or heads. Location is another big one, like the temporalis muscle near the temporal bone. And sometimes the name tells you the origin and insertion points where the muscle attaches. Sternocleidomastoid attaches to the sternum, clavicle, and mastoid process.
It's not just random names then, it's a descriptive system.
It's a very logical system once you learn the terms. It helps you understand the muscle's likely function and location. And remember how muscles work together. The main mover is the agonist. The muscle opposing it is the antagonist. Synergists help the agonist, maybe adding force or stabilizing joints. And fixators are synergists that specifically stabilize the origin bone of the agonist, providing a firm base.
And muscles always pull, right, from insertion towards origin.
That's the rule. They pull the insertion point closer to the origin point. Oh, a quick side note on smooth muscle we mentioned earlier.
Yeah.
It also uses actin and myosin, but the trigger is a bit different. Calcium comes in, binds to a protein called calmodulin, which then activates an enzyme that allows myosin to bind actin. No troponin involved, just a different regulatory pathway for those slower, sustained contractions.
Okay, fascinating detour. So we've covered the muscles, the engines. Now let's talk about the command center, the system pulling the strings, the nervous system.
Right. The nervous system. Its basic job is threefold. Receive sensory input, integrate or process that information, and then generate a motor output or response. It does this using those core properties we mentioned for muscle too, but applied differently. Excitability, responding to stimuli, conductivity, transmitting signals, and secretion, releasing neurotransmitters.
And functionally, it's like a pathway, input, processing, output.
Exactly. Sensory nerves, called afferent pathways, bring information in towards the central nervous system. Inner neurons, mostly within the CNS, do the processing and decision-making. Then motor nerves, the efferent pathways, carry commands out to muscles or glands.
And the main anatomical parts are the CNS and PNS.
Correct. The central nervous system, CNS, is the brain and spinal cord, the integration hub. The peripheral nervous system, PNS, is everything else, all the nerves branching out, connecting the CNS to the rest of the body. The PNS itself divides into the somatic system, which controls voluntary muscle movements, and the autonomic system, handling involuntary stuff like heart rate, digestion, breathing.
And this vital CNS needs serious protection.
Oh, absolutely. It's heavily protected. First layer. Bone, the skull protects the brain. The vertebral column protects the spinal cord. Underneath the bone are three membranes, the meninges, the tough outer dura mater, the web-like arachnoid mater, and the delicate inner pia mater that clings to the brain and spinal cord surface. Between the arachnoid and pia mater is the subarachnoid space, filled with cerebrospinal fluid, CSF. This fluid acts as a shock absorber, cushions the brain, and helps with nutrient waste exchange.
So layers of bone, membranes, and fluid. Okay. Now, the basic building block of this whole system is the neuron, right? The nerve cell itself.
That's the one. The neuron is highly specialized for transmitting signals. It has several key parts. Dendrites are typically short, branching extensions that receive signals from other neurons. Think of them as the input antennas. The cell body, or soma, contains the nucleus and organelles. It's the neuron's metabolic center. The axon hillock is where the axon originates from the cell body. It's often the trigger zone where the decision to fire a signal is made. The axon is a long projection that carries the signal away from the cell body, sometimes over very long distances.
Like the main transmission cable.
Exactly. Axons are often insulated by a myelin sheath, a fatty covering that speeds up signal transmission. There are gaps in this sheath called nodes of Ranvier. At the far end, the axon branches into telodendria, ending in synaptic end bulbs or terminals. These bulbs contain the neurotransmitters, and the whole junction where one neuron communicates with the next cell is called the synapse.
So that's the main player, the neuron. But they don't work alone, you said. There's a support crew.
Yes, the neuroglia, or glial cells. They're often called support cells, but they do much more than just support. They are essential for neuron function and health. There are different types in the CNS and PNS.
Okay, who's in the CNS crew?
In the CNS, you have astrocytes, star-shaped cells. Very numerous. They help form the blood-brain barrier, regulate the chemical environment around neurons, provide nutrients, and more. Super important. Oligodendrocytes are the cells that produce the myelin sheath around axons within the CNS. One oligodendrocyte can myelinate multiple axons. Microglial cells are the immune defenders of the CNS. They act like phagocytes, cleaning up debris and fighting infection. And ependymal cells line the cavities of the brain and spinal cord and help produce cerebrospinal fluid, CSF.
Quite the team. And in the PNS?
In the PNS, the main players are Schwann cells, which form the myelin sheath around peripheral axons, unlike oligodendrocytes. One Schwann cell myelinates only one segment of an axon. And satellite cells surround neuron cell bodies within PNS ganglia, providing support and regulating their environment, somewhat similar to astrocytes in the CNS.
So these glial cells are absolutely vital. Neurons couldn't do their job without them.
Not effectively, no. They are critical partners in nervous system function.
All right, so we have the cells. How do the signals actually travel? We talked about action potentials earlier with muscles. Are they the same in nerves? And what about graded potentials?
Good question. Both occur in neurons. Graded potentials are small, localized changes in membrane potential. Think of them as ripples in a pond. They happen usually at the dendrites or cell body when a neurotransmitter binds. Their size depends on the strength of the stimulus, and they die out over short distances. They don't travel far.
So like whispers that fade quickly.
Exactly. They can, however, add up, and if they're strong enough to reach the axon hillock and trigger a certain threshold voltage, then they can initiate an action potential. Action potentials are different. They are large, rapid reversals of membrane potential, an all or none event, like flipping a switch.
The full-blown shout that travels the distance.
Precisely. They are generated only by muscle cells and neurons, specifically at the axon hillock and along the axon. Crucially, they do not decrease in strength as they travel down the axon. They regenerate themselves along the way. This allows signals to be transmitted reliably over long distances, like from your spinal cord all the way down to your toe. They are the primary means of long-distance neural communication.
And you mentioned myelin helps them travel faster, saltatory conduction.
Yes, saltatory conduction. In myelinated axons, the myelin sheath acts as an insulator. The action potential doesn't happen along the myelinated parts. Instead, the electrical current flows rapidly under the myelin from one node of Ranvier, those gaps in the myelin, to the next. The voltage-gated sodium channels needed to generate the action potential are concentrated heavily at these nodes. So the action potential effectively jumps from node to node.
Like skipping stones across the water instead of swimming the whole way.
Perfect analogy. This jumping is much, much faster than continuous conduction along an unmyelinated axon, where the action potential has to be regenerated at every single point along the membrane. Myelination dramatically increases nerve signal speed.
Okay. And electrically, what's happening during that action potential jump or shout? You mentioned polarized, depolarized.
Right. So at rest, the neuron membrane is polarized. There's an electrical difference across it. It's more negative inside than outside. This is the resting membrane potential, maintained mainly by sodium-potassium pumps.
Ready and waiting.
Exactly. When a stimulus triggers it past threshold, voltage-gated sodium channels fly open. Sodium ions, Na+, rush into the cell down their concentration gradient. This influx of positive charge makes the inside of the cell rapidly become positive. That's depolarization.
The signal firing.
Yes. But almost immediately after, those sodium channels close and voltage-gated potassium channels open. Now, potassium ions, K+, which are concentrated inside, rush out of the cell. This outflow of positive charge makes the inside of the cell become negative again, returning toward the resting state. That's repolarization. There's often a brief hyperpolarization where it becomes even more negative than rest before settling back.
So it's a rapid wave of ion movement, sodium in, potassium out, that travels down the axon.
That's the essence of it.
Huh.
A very quick, precisely controlled sequence of opening and closing different ion channels, some opened by chemicals, some by voltage changes, voltage-gated, and some even by physical force, mechanically gated.
And these neurons need to pass the message along
Like
the
this-
synapses, right?
Correct. The synapse is the junction where information is passed from one neuron, the presynaptic neuron, to the next cell, the postsynaptic neuron, which could be another neuron or a muscle or gland cell. There are different configurations. Axodendritic, axon to dendrite is common, but also axosomatic, axon to cell body, axoaxonic, axon to axon, even dendrodendritic.
A whole network of connections.
An incredibly intricate network. And the signal passed across the synapse can either be excitatory, meaning it makes the postsynaptic neuron more likely to fire its own action potential, or inhibitory, making it less likely to fire. This allows for really complex processing, weighing different inputs before deciding on an outcome.
And sometimes the message needs amplification or a more complex response inside the cell. You mentioned first and second messengers.
Yes, it ties back to cell communication. The neurotransmitter crossing the synapse is the first messenger. It binds to a receptor on the postsynaptic cell. But sometimes that binding doesn't directly open an ion channel. Instead, it triggers a chain reaction inside the postsynaptic cell, often involving a second messenger. Common second messengers include cyclic AMP, NC-AMP, or cyclic GMP, CGMP. These molecules then go on to activate enzymes or influence gene expression or open other ion channels indirectly. It's a way to amplify the signal or trigger more complex, longer-lasting changes within the cell beyond just a quick electrical blip.
Wow. Okay, let's try and tie this all together for everyone listening. It's quite a journey. So the nervous system kicks things off with an action potential, that electrical shout. It travels down a neuron, maybe jumping along thanks to myelin.
Right.
Reaches the neuromuscular junction. The arrival triggers extracellular calcium to rush into the nerve ending, causing it to release acetylcholine, the first messenger.
Yep.
ACH crosses the gap, binds to receptors on the muscle, opening channels for sodium to rush into the muscle cell, causing depolarization there,
Creating the muscle action potential.
which travels into the muscle fiber and tells the sarcoplasmic reticulum to release its intracellular calcium stores.
The second crucial calcium role.
That calcium then binds to troponin, which pulls tropomyosin out of the way,
Unlocking the actin binding sites.
allowing the energized myosin heads to grab onto actin, pull, release, and repeat, using ATP, the sliding filament theory, in action, shortening the muscle.
You got it. A beautiful cascade. From nerve impulse to muscle contraction. System working together.
It really makes you appreciate just how complex and, well, elegant our bodies are. Every single movement, every thought relies on these incredibly intricate processes happening perfectly, constantly.
It's a silent marvel happening billions of times a second within us. Understanding even the basics gives you a real sense of wonder about it all.
Absolutely. And maybe a final thought for you, our listener, to ponder. Given how precise this whole chain reaction is, what happens if just one tiny piece malfunctions? A faulty ion channel, maybe, or damaged myelin, or not enough neurotransmitter. Think about the potential ripple effects on movement, sensation, even thought. And how might science even begin to address those issues within such complex biological machinery?
It's a profound question. It highlights the fragility alongside the resilience and drives so much medical research trying to understand and intervene when things go wrong.
Well, thank you for joining us on this deep dive into the amazing world of muscles and nerves. We hope it sparks some curiosity.
Definitely. Until next time, keep diving deep.